Hi, so we're going to be talking about the resting membrane potential and action potentials. So we've been talking about cells, and we've been talking about how there are different electrolytes or ions on one side of the cell versus the other, namely sodium on the outside, potassium on the inside. But we've also talked about how the distribution of charged molecules, including proteins, has created a minus 70 millivolt difference of the inside. So the inside is around minus 70 and the outside is positive. So we can see this just experimentally. We can, this has been done many times. You put an electrode on the inside of the cell versus the outside, and you can see this charge differential. Okay, that comes to about minus 70, between minus 70, minus 90. It depends on the cells, but all cells will show this this negative potential on the inside. So this is primarily, like I said, from charged molecules or ions. And we can kind of calculate what this is going to be. So this is something called the Nernst equation. It measures the membrane potential, and you can calculate that by the distribution of ions. And there's something called the Goldman-Hodgkins-Katz equation, which is kind of an extension of this, that can take in every molecule or every ion that's inside the cell and that concentration, and it will spit out a calculation of what that potential is. Now, we don't have to calculate the membrane potential across a cell, but using this equation, we can do that for individual ions, and we can say, well, if it's the concentration is this on the inside versus the outside, we can calculate that and determine what the potential is. Now down here we have an equilibrium potential. Now that's a little different and can be kind of not really confusing. It's not something we have to calculate, but it helps to really understand what it's saying. And in a typical cell, we can look at that and we can say, so I have down here marked, I have the equilibrium potential for potassium. That's the one I have pointed to. It's got all of these on there. And what this is saying is that based on the ion concentration of cells, so this is including everything, calcium, proteins, DNA, potassium, chloride, everything, based on all of those and their basic distribution within and outside those cells, this says that potassium will equilibrate, and in other words, neither go in nor out of the cell when the inside of that cell is at minus 90, okay? So we already know that potassium is a high concentration inside the cell, okay? Well, it's part of the reason potassium is okay with staying inside a cell is because it's negatively charged. Normally, potassium would go down its concentration gradient and try to leave. Well, this is saying that minus 70, which is where a cell typically rests, maybe isn't negative enough for potassium, so it's still really trying to leave most of the time. And it will continue to leave until it's minus 90 in there. Now, this has a physiological use, and that is that if we want to really hyperpolarize a cell, all we have to do is really open it wide open to potassium, and potassium will leave that cell until it's minus 90 inside the cell. Okay. And then it'll stop leaving. It'll be like, okay, I like this concentration difference. I like the fact that it's minus 90. It's a lot more negative in here, and I'm a positive ion. That's where it's going to be satisfied. That's where it's going to reach an equilibrium. Is when it's minus 90 in the cell. But as long as it's above that, the potassium is going to be leaking out. And that's something else we're going to talk about, is these leak potentials. Now, if we look at some of these others, chloride is actually happy exactly where it is. It says, okay, you know what, it's negative on the inside, have no real interest in going in there, but there's a lot of chloride out here, so I kind of have an interest in going in there. And its equilibrium, where it won't move in or out, or it moves both, but it's at equilibrium, is at minus 70. Sodium just wants to go in. Okay, sodium, it's until the inside of that cell is a positive 60, it's just going to keep going in. Calcium, even more so, it's going to go in until it's a plus 130. Okay, so that's all that's doing, all this graph is telling us is that at what potential these things will be at equilibrium and whether or not they want to go in or out. Okay. So the membrane potential, there are a few different points, I guess, to make before we really get into it. Electrochemical. It's electrochemical. Now, electrical usually just means you're talking about electrons. You know, it's just negative charges or a charge. Electrochemical means that we're basing it on chemicals. So it's charged molecules, or in this case, charged ion, which isn't a molecule. It's an atom that's it's an ion. It's a charged like a charged sodium or chloride that's just been ionized and is maintaining its charge. So that's what we mean by electrochemical. The resting membrane potential, that's the voltage at rest, when the cell isn't doing anything, and by at rest we mean it's not depolarizing. It's like if it's a neuron, it's not active. If it's a muscle cell, it's not contracting or depolarizing for any other reason. So that's when it's at rest, and that's what we talk about, most of them being at about -70. So we're going to talk about how that happens. And then the action potential. The action potential is when it depolarizes and repolarizes, and then that's a signal that's passed down. So when a cell, such as a muscle cell or neuron, rapidly changes potential, okay, so depolarizes, and then repolarizes back to that resting membrane potential, that's an action potential, okay? And then I have this last comment that I've said a few times, that it's usually about -70 inside of the cell. All right, so maintaining that resting membrane potential. One of the big factors in maintaining resting membrane potential is the sodium-potassium pump. Okay, so we've talked about that, the sodium-potassium ATPase, and that's going to push out 3 sodiums while it pumps in 2 potassium, right? So you can see that already, if this is your cell membrane, you can already see that if we're pumping out 3 positives and only pumping in 2 negatives, that's going to contribute to the negative charge of the cell. Now, we also mentioned that potassium kind of wants to leave. Okay, until it's -90, that potassium will want to leave, and it's possible. There are leak channels that allow it to go, that allow it to leave, which ultimately what happens is you kind of have this dynamic sort of homeostasis, that's what that word is for, where you're pumping the potassium out, or you're pumping the, oh, I put that totally in the wrong direction. Yeah. So you're pumping the sodium out, and you're pumping the potassium in while the potassium is also leaking out. But it's maintained to that point where it's at a -70. So the main factor then is that potassium. Here, I'm going to make that point on this slide. Potassium is the main factor in that resting membrane potential. Okay. So potassium is constantly leaking out, but also continuously being pumped back in by the sodium-potassium pump. And when there is no net change, that's where we're at resting membrane potential. Okay. So high or low potassium levels will change the potential. So if you have a lot of potassium, if you have what we would call like hyperkalemia, then what's going to happen is that the potassium is not going to want to leak out as much. If the potassium doesn't want to leak out and take that positive charge outside with it, then it's going to become more of those positive ions are going to stay in there, and it may go from -70 millivolts up to like -65, and then it may depolarize completely at like -50 if it gets that high. So when your potassium levels are really, really high, then the inside of that cell becomes less negative. It kind of starts to depolarize. And of course the opposite is true as well. If your potassium levels are low, then that potassium is going to want to leave the cell even more, which is it's going to remove that positive charge from inside the cell, and it's going to make it more negative, which is going to hyperpolarize that cell. Okay, so that's why potassium levels are really important. That's why we measure those with like a metabolic panel, and we can see your potassium levels, sodium levels, and we need to correct those if they're out, because that can cause the heart to do some funky things. Okay, so this keeps the membrane at a steady resting membrane potential. Sodium can also leak in, but it's not as permeable. There aren't as many of these sodium leak channels as there are these potassium leak channels. Okay, and that's what gives potassium that power to maintain that resting membrane potential. Okay, so this kind of takes you through the whole process, kind of everything we just talked about. All right, so we're going to apply this now to nerve function and an action potential. So let's first look at the structure of a neuron. So a neuron has the dendrites. Dendrites is where they receive signals. So if we have another neuron up here, okay, with the cell body and dendrites, all right. So this is how neurons communicate. They communicate from the axon terminal, which is this little bulb at the end, and then they will communicate with the next neuron at its dendrite. So dendrite is the incoming signal, and then that message can travel through the cell body, and then if it reaches this, what we call the axon hillock, then an action potential can move down the axon to the axon terminal, okay? And then that will send a signal to the next one, okay? The next neuron, maybe it's dendrites. This is a terrible picture. And then it just kind of continues. And I have another picture of this that's slightly more clear than what I just drew. All right, so I did want to say something about something called dendritic spines. So on these dendrites, there are little spines that kind of increase the surface area and increase the potential of contacting other neurons. So if you have a neuron that's trying to communicate with these dendrites, so if this is the dendrite, these little guys here are little spines on the dendrite. These are the dendritic spines. If you have a neuron that's trying to communicate, well, if you have more of these spines, then that's more area for communication. So you can form more synapses, which means that you can exchange more information or store more information. And this is kind of why I'm pointing this out, because there have been studies done that show that the increase in these spines is associated with learning and memory. And so if you're trying to memorize something or learn something, kind of going over it over and over, figuring it out, then what happens, and usually it's shown to happen more in the sleep, during sleep, is that you will grow more of these spines. You can't increase or decrease the number of neurons in your brain, not really, but you can increase the connections that they make by sort of growing out these dendritic spines. So it's really interesting and useful. Here's some more pictures of neurons. They're very pretty. Here you can see what was added are myelin. So there are myelin sheaths that myelin also written here. Myelin sheaths that kind of increase the speed of transmission that we'll also talk about. All right, so let's talk about synapses in general. We're not going to get into the detail we'll get in later, but that's how neurons communicate with each other. A synapse is not a connection between two neurons, but it's a space. You can see here, this is kind of blown up. You can see here that there's a space between these. And that's going to have to do with the chemical nature of transmission. So we might release, so if we have a neuron here and then it's coming down to a spine down here, then you may release little molecules, little neurotransmitters that will then bind to receptors on the other side. So that's how communication in neurons takes place. You can have one neuron that communicates with a whole bunch of other neurons, okay, and send signals all over the place, or you can have a whole bunch of neurons, a whole bunch of neurons that may communicate with one. Okay, so you've got lots of options, and it really has to do with where they are and what kind of information is being processed. All right, so those are kind of synapses. They can be inhibitory or excitatory, and we'll get into how that takes place too. Okay, action potentials and gated ion channels. So now we're going to start talking about these action potentials. So some terms that I'm going to go over a couple of times. We need to tell a channel when to open or activate a cell. So we've already figured out that the inside of the cell is negatively charged and that sodium is a high concentration on the outside. To depolarize that cell or to change that membrane potential to a positive, so we're kind of looking at something like this, all we have to do is open it up to sodium, and that will depolarize the cell. Okay, and that's what's going to trigger and get this action potential going. So that's your depolarization. So it occurs when positive ions rush into the cell, changing the membrane potential from a negative on the inside to less negative or even positive, like all the way up to like a plus 30 or something. So it goes from minus 70 all the way up. So how do we do that? Well, there are gated ion channels. So gated ion channels just mean they open and close because of some sort of stimulus. Here we can kind of see that down here. We can see this channel is closed and the sodium is kind of stuck on the outside. But then when the channel opens, then sodium can rush in. Okay. We'll also see later that that potassium will rush out, not necessarily in the same one, but we can see that when these are open, they're open to they're usually very specific. So ligand gated just means that you put a receptor on it. So that's really what this photograph is kind of showing you. There's acetylcholine. When acetylcholine binds, acetylcholine will act as a ligand. A ligand is just something that binds to a receptor, some kind of particle that binds with a receptor. So when the acetylcholine binds to it, as we can see happening over here, then that's when the channel opens. Okay, and it can be permeable to whatever that channel happens to be permeable to. So that's the example. Acetylcholine binds to an acetylcholine receptor, and the channel opens, and then sodium enters the cell and depolarizes it. So that's what we can imagine happening right here. Okay, so this cell here, an action potential moved down that neuron, and then it caused acetylcholine to be released. Acetylcholine binds to a receptor, an acetylcholine receptor, opens a channel, and sodium rushes in. Now the red here shows depolarization. Okay, so if we could imagine that the stimulus takes place at this point, and this is where we have that initial depolarization. So we can see that it's very intense here, maybe plus 30 depolarization. So it went from minus 70 to plus 30. But as we move farther away, you can see this red color kind of fades. Okay, so that signal is getting weaker as it moves away, less intense as it moves away from this area, because this is where the sodium is rushing in, right there, where the neuron is, where it sends the signal, and where that channel opens. So you can see that it weakens over distant. We call that a graded potential. Grading is in, like, if you grade something, you give it a range of values. But it weakens. And you can kind of see this happening here, the same kind of thing, that area of depolarization. So how do we get it to stay depolarized, or how do we get it to depolarize completely? Because what we want to ultimately do is get the signal down to this area, which is the trigger zone. Okay, and we'll talk about what happens to the trigger zone. There's a hint. There are voltage-gated channels down there that are waiting to see if you can get this depolarization signal all the way down there. And then if they do, then they will be alerted and they'll send out the action potential. But right now, we just have to get to that point. We have to get this area depolarized. Well, in this picture, we see this nice little picture where we can see maybe a depolarization event trying to happen here and a depolarization event trying to happen here, but then it's interrupted by an inhibitory event. So this is excitatory, so these reds are excitatory. Could have made that in red. And this down here, the blue, is inhibitory. All right. So maybe we didn't get a signal. Okay, so we had two summing together. This is spatial summation because they're in two different places that arrive at the same time. So that's spatial summation. But then that inhibitory signal kind of messed everything up, and maybe we released chloride into the cell. Remember, chloride is going to come in and try to keep it at -70. So maybe we put some chloride, negatively charged chloride into the cell, and it caused it to not depolarize. Okay. So spatially or temporally just means that you have a whole bunch of signals at one time. I've got a slide on it later. So the excitatory signals are caused primarily by an influx of sodium, promoting depolarization. Inhibitory signals are primarily caused by the opening of chloride channels, which inhibits depolarization, or yeah, that's inhibitory. And here we can see a bunch of them. We can see here's an excitatory, so plus, plus, plus. And then we see one little negative down here. So what we're trying to do is get that signal, that depolarization signal, to happen here at the trigger zone. All right. So the trigger zone, or the axon hillock, that is where everything's going to kind of tell the story. So we've talked about a couple of terms here, a couple of more. Depolarization. When we're talking about an action potential, depolarization is that initial sodium is coming in. Okay. But then we don't want it to stay that way. So what happens is potassium is allowed out almost immediately, and you have repolarization. So this is polarized when it's at -70. Depolarized, we call it depolarized when it moves up to +30 millivolts. Or so, you know, around, whatever it happens to be. So that's depolarization is in this area, and I have other graphs that kind of show this. And then repolarization is in this area. And really, kind of the way I drew this, it looks like it goes right back down to resting membrane potential, but it actually kind of goes down all the way and hyperpolarizes. Okay, so this is hyper polarizes, so it actually polarizes. So it actually goes below -70 for a little while, okay, before it finally levels out back at that resting membrane potential. Okay, so depolarization, it's from the resting membrane to less negative or positive. Repolarization, it kind of goes back down, approaching that resting membrane potential. And then hyperpolarizing means that it's gotten even more negative. So it's more negative, like maybe down to, you know, minus 80 or something. And remember, we already said that if you open it up to potassium, which we did here, if you open it up to potassium, potassium's not going to be happy until the inside is -90. So that's what contributes to that. All right, so this is the graph that I just tried to draw. So we have the inside of the cell is -70. Sodium wants to get in. Depolarization. Sodium rushes into the cell. We allow that to happen at this axon hillock, and then potassium will rush out and repolarize it. Okay, and those are just your basic questions. What causes depolarization? Sodium rushing in. What causes repolarization? Potassium rushing out. Okay, so this action potential, this event that we just talked about of the depolarization, repolarization, and then hyperpolarization, and then returning to resting membrane potential, that can happen in 1 to 4 milliseconds. This whole entire event may happen hundreds of times per second, a couple hundred times per second. Okay, that's how quickly this takes place. So that's kind of what this is showing you. So there's 25 milliseconds, which is a quarter of a second. Or less than that. And you can already see how many of these action potentials or these depolarization events can happen. So this ability is possessed by neurons, muscle cells, and then there are some other kinds of cells that can also participate. So how does this do this? So we want to get into this thing that happens. We kind of talked about the graded potential as it moves across the membrane, that depolarization event. So we've got excitatory potential, so depolarization events that happen on the, let's see, so what I'm drawing here is kind of an axon or a neuron. Okay. Remember, it's moving down here to this, and we'll just make it short, to this trigger zone at this area. So we've got the depolarization event. Maybe this one depolarized, this one depolarized. Let's say they all did. They're all trying to depolarize this cell. It's unanimous. We've decided we're going to depolarize this cell. Well, when we get to that axon hillock, the axon hillock, something has to happen, okay? Because we want that the action potential is actually going to occur down the axon. Okay, so this was a graded potential. It was fading, weakening, and action potential doesn't do that, and we'll get into why that's the case. But at this point, to sense that we want this cell to start an action potential and to send that signal, there are voltage-gated channels down there. So voltage-gated sodium channels. So they're going to detect that there has been a depolarization event that takes place. And that's what these guys, this is kind of what this is showing a picture of. But really, all it has to do with there are charged or polarized amino acids sitting in the inside of this, or sitting on this protein, that it changes the configuration so that it opens up when a potential difference happens, when a charge difference happens. So, so the voltage gated, they open or close in response to small voltage changes across the cell membrane. At rest, it's negative on the inside. When the cell is stimulated, that relative charge changes and the voltage gated channels open or close. The most common voltage-gated channels are sodium and potassium. We'll also see calcium channels. We see these in cardiac. We see them in cardiac muscle, skeletal muscle, smooth muscle, nerves. We see them in any kind of cell that's electrically active. Okay, so this is kind of the whole process of what we're looking at here. We've managed to get our signal down to this trigger zone, this axon hillock. We've had enough depolarization from the ligand-gated channels to let the sodium in to depolarize it. And now we get into that trigger zone. And we open up some of these voltage-gated channels. Okay. So we open up the voltage-gated channels, which is going to cause sodium to rush in. So what we've done is we've already said, okay, there's a bit of a positive charge here, but it's fading. But it gets there and it says that's enough. Okay, so let's say it's over minus 50. It's depolarized enough that it activates these voltage-gated channels. They open wide up and they let sodium in, which is going to cause a huge depolarization in this event, in this area. So we've really depolarized the cell now in this area, in this trigger zone. Well, there's another voltage-gated channel sitting right here that says, whoa, what happened? We just got depolarized, and it's going to open up to potassium or sodium, and it's going to depolarize too. Okay, and then there's another voltage-gated channel here, and it says, whoa, what happened? And it's going to open because of the depolarization. And it just keeps going. It depolarizes here, which causes this one to open. It depolarizes here, which causes this one to open all the way down. And that's really what an action potential is. One thing we do know is that we can't leave them depolarized, so we're going to have to fix that. But the overall concept of what an action potential is, it is this series of depolarization, repolarization events. This picture up here is showing dominoes. So we have something called an all or none principle. So if we kind of go back here to the axon hillock and we say, okay, there's these initial, in this trigger zone, there are these initial voltage-gated channels that open, then that's going to cause the the action potential. But that's more like pushing over the first domino in a series of dominoes. Because after that, it's just a click, 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 click all the way down. It's not going to become more intense, and it's not going to weaken as long as it's moving across this area. So that's the concept of the domino. Is it doesn't matter if you hit that first domino with a truck, you know, if you hit it as hard as you can, it's not going to matter. The rest of these dominoes are going to just fall like they would have fallen no matter what. You can't make an action potential signal down the axon stronger or faster just by giving it a bigger hit from the beginning. It just goes. It just goes and does what it does. Okay, so now, no matter how strong the stimulus, as long as it is greater than the threshold, the action potential will occur, and that's it. So that's what this is showing too. 1, 2, 3, 4, 5, all the way down. This just shows some other pictures. So the resting. So this is your resting membrane potential, activation of the voltage-gated channels, activation of ligand-gated channels. So that's just kind of showing the different kinds of channels here. So the question then, if we go back to look at this and we say, okay, it's moving in this direction, similar to dominoes. So if we start the signal at this point, we knock over that domino can't be knocked over again. It's already depolarized. It's done. So that means that keeps things moving in this direction. However, in the body, we're setting these dominoes back up again. We're getting them ready for their next round. But that takes a while. So we've got this thing called the refractory period. So if we have, let's see, let me draw this different. All right, so here we'll just do it like this and like this. All right, I don't know if this is going to make any sense. That should be open. But if we say sodium rushing in is causing depolarization, so that's sodium rushing in, and then immediately after that, potassium rushes out. Whereas when we started this process, sodium was high on the inside and potassium was high on the outside, or potassium was high on the inside and sodium was high on the outside. When we're done with this depolarization event, we flipped everything, okay, because the potassium has moved out. That means for just an instant, you can't depolarize that cell again. So if we were to try to go backwards, if we have depolarization that happens here, immediately when that happens, sodium rushing in, potassium rushing out, at this point right here, everything's in the wrong place. The sodium is on the inside and the potassium is on the outside, which means we can't have another action potential at that time. We have to pump everything back to where it was before we can have an action potential. Well, in the meantime, that depolarization has triggered another voltage-gated channel to cause depolarization here at number 2. Okay? So number 2 depolarizes, and it's going to send out a signal, of course, in both directions, but it doesn't matter because this one's already depolarized. However, 3 hasn't depolarized yet. It's ready to go. So now 3 is going to depolarize. And then 4. Well, by the time it gets down to, 4 or 5, I don't know, we're just using this as an example, by that time maybe 1 will be ready to go again. But 3 won't be. Okay, so it's going to keep it moving down this direction because of what we call a refractory period, and that's what this is showing. So we have an absolute refractory period when we have the sodium rushing out, or the sodium rushing in and the potassium rushing out. Okay, so this is sodium in, potassium out. Okay, and everything's in the wrong place. So that dark purple means that it's backwards. It can't depolarize again. Now we have this pink area. We call that a relative refractory period because if a signal is strong enough, it can cause depolarization. It can cause it to once again let the sodium back in. but it kind of has to get below, get below that threshold. If it's still above the threshold, it can't happen. And the threshold's usually, I don't know, usually about -50. -50 is kind of that cutoff where the voltage gated channel says, okay, let's open. So if it's still above that in this area, there's no chance that it's going to open again. If it gets below that, it'll open again, but there may not be very much sodium to cause a big signal. So this pink area is really where the sodium-potassium pump is kind of doing its thing, and it's trying to get everybody back into order. It's saying, okay, sodium, you need to go out, potassium, you need to go in, and it fixes it. It happens very quickly. It's kind of amazing. So that's your absolute refractory period, and really it's the absolute refractory period that keeps it flowing in that one direction. So you've got your absolute because it's above that threshold. Once it gets below that threshold, then you might be able to get another action potential out of it, and then you're back to resting membrane potential, and that's when you're really ready to go again. Okay. So let's see, the refractory periods are also the key in determining the direction of the action potential, so that's going to be, that's important to know. It limits the number of action potentials that can happen. So when you have what we would call like a tetanus, like the disease tetanus, but really a tetanus is repeated depolarization events, activation events. Those are limited. So most neurons respond at frequencies of up to 100 action potentials per second. And for brief periods, maybe a little higher, but that's it, and that's because of that refractory period. It's just not constantly always ready to go. It has to pump everything back to where it was. All right. So transport across cell membranes. These are all kind of just reiterating what we've already talked about. The active region. So we get to that axon hillock. Remember, we have the sodium. Are the voltage-gated sodium channels in this area, and we have depolarization. It becomes positive on the inside, negative on the outside, and then that just keeps moving down the axon. Okay, and it keeps going without increasing or decreasing in strength. Same thing here, shows the depolarization overshoot, so we've gone all the way up to +30. And then hyperpolarizing because it went down too far because we're letting all that potassium out. And then everything closes, gets pumped back into place, and you're back to resting membrane potential. Okay. All right, so a couple of points here to be made. This graph is kind of nice because it breaks everything down. It shows the depolarization phase, the repolarization phase of potassium rushing out, and then how it's spreading, but it can, everything that we just kind of went over, it just breaks it down into individual steps. So yeah, action potentials are unidirectional. They can only go that one direction because of the refractory period. The velocity propagates along the membrane, depends on the fiber diameter. So a small diameter fiber, okay, I can't even really draw anything in here because I've made it so small. But if you have molecules, charges, barely perceptible here, you've got so much area that's like right there at the membrane that signal can leak out of the membrane easily. So I know we talk about how the membrane is semi-permeable, but it can leak out. It's just phospholipids, and while those are good, they're not going to prevent any kind of, anything from, sneaking out. So if we have larger diameter, then these sodium, so I'm going to say the red is sodium, you've got those that are going to move along through that neuron kind of in one direction or the other. Well, we'll just say it's one direction because we know that's how it works. But we can see that the sodium is moving along while some of it is leaking out. Okay, and so that signal is fading, and that's why we need an action potential, and we can't just like open it up in the very beginning and say, okay, sodium, go in and go all the way to the neuron that happens to be 3 feet long because it's a neuron that's a sensory neuron in our big toe. That signal is not going to make it that entire distance. So larger means that you have proportionally less leaking out. So yeah, you're going to have some leakage, but you've got more inside. Meanwhile, if it's smaller, then your proportion is you're going to have most of it leaking out quickly. So that's why a larger diameter works better. But, which is what the second thing says, it depends on the fiber diameter and whether or not the fiber is myelinated. Okay, so that's our point. Larger the fiber diameter, the faster it's going to go. Current is lost to the membrane if there's less membrane compared to the volume, but we're going to myelinate them. So let's talk about myelin. Myelin is a fatty covering that electrically insulates axons, making action potentials move more quickly down the axon. So let's kind of try to draw this in 3 dimensions here. Okay. Made it red and I didn't really want to. Yeah, I'm going to redraw it. All right, so this is our axon. And we see that there's some leakage out, okay? So we've got sodium that is going to move as a current. So if you think about how electricity runs through a wire, it's just these electrons that are just zipping through the wire, because that's current flow. And sodium also can flow through current flow. It's flowing in a liquid substance, but it's going to flow pretty quickly through that. So that's current flow. And then we have some leaking. Okay, so this is why it fades over distance, because some of that's leaking out. We don't want it to. We try to stop it, but you know, what can you do? What myelin does is myelin will coat it and prevent it from leaking. Okay, so if we put myelin here, then it doesn't leak out. Now, one thing I didn't mention actually is that while I said that current flow is very fast and the sodium will go down, an action potential doesn't work that way. An action potential is depolarization, repolarization, depolarization, repolarization, all the way down. Depolarization. It's fast, but it's relatively kind of slow. Okay. Current flow, by comparison, is very fast. So if we can get rid of some of this depolarization and repolarization, and just coat it with myelin, and let that current flow kind of do its thing, then we have this whole area that's very fast. Okay, so we've got this fast current flow that's happening here, and then if it starts to fade, which it will, then we can just open up a voltage-gated sodium channel here and repolarize it again. So if that signal, as it goes along, it's very strong here, very strong, but then it fades, then we can strengthen it again at the nodes along the way. So we're going to look at this again in the next picture, and it's going to be a better picture than that I just drew, but hopefully it'll make sense. So Schwann cells, or myelin. Myelin is laid down by Schwann cells, a certain type of cell called Schwann cells in the periphery, and oligodendrocytes in the central nervous system. We'll come back and talk about that, I think, when we talk about neuro, officially. But anyway, this myelin kind of surrounds these nerve fibers in that way. All right, so let's look at this. I told you it's a better picture. So here we have sodium depolarization, sodium rushing in, depolarizing the cell, and then we can have that positive signal. Here, I'm going to make it red because it's easier to see. We have that positive signal that starts out very strong, very intense, because we just let it all in, we let all that sodium in, but we stop the action potential, so as it moves along, that signal is going to fade. So it starts out okay, but then it starts to fade. So even though the myelin is preventing that loss, It's still going to happen a little bit. It's not going to insulate it perfectly. Okay. Well, then we have to recharge it again. Okay. So now we're going to let more sodium in. So we get a big signal here again. That's going to fade. And then we get to this point and we let sodium in again, and we get a big signal again. That's going to fade. All right, so that's basically what myelin is doing. And this current flow that happens here, I'm going to erase all that. If I said the action potential is slow, depolarization, repolarization, depolarization, kind of like that, under this myelin, you just have current flow that's very fast. So it's fast, and then it's depolarization, and then fast, fast. Okay? So you have that current flow under the myelin that's happening very quickly. The more myelin you have, the longer those tracks of myelin are, the more current flow you have, the faster the potential. Okay, so this is why you're able to have a neuron that is 3 feet long that goes, you know, from your spinal cord all the way down to your big toe. Why you're able to have that kind of a signal and walk and actually feel something, like if you step on something, you can feel it right away. It's because we have myelination. If we didn't have myelination, to get something to travel that fast, we would have to have huge, like, you know, I don't know, a whole centimeter in diameter neurons that are passing the signals along. Okay, so our anatomy here, this is the myelin. The nodes at which we have depolarization, or that recharging area, those are the nodes of Ranvier. It's French. Nodes of Ranvier, and that's where those are taking place. Okay, so let's talk about a couple of things. The generation of action potentials is prevented by local anesthetics such as procaine, lidocaine. So these drugs block voltage-gated sodium channels. Now this should make sense. If our sensory neurons are in our skin, if we have all of these sensory neurons that are being passed with action potentials, if we inject in this area, this is a syringe now. I didn't know I was drawing that. It looks like a knife. But if we inject some lidocaine into this area and it blocks those voltage-gated sodium channels, then we have no signal. Okay. And that's the way those drugs work. They block voltage-gated sodium channels. And they block them for, you know, as long as they do. Now, this is not something that we would be able to take orally. You can't take a local anesthetic orally. You don't want to shut down all of your voltage-gated sodium channels in your body. You would die. So, right. So without action potentials, graded signals generated in the periphery in response to injury, for example, cannot reach the brain and give rise to that sensation of pain. It's not going to move through. Even if it's myelinated, you still have the voltage-gated channels that are passing that signal in the nodes between the myelin. There are toxins. Some animals produce toxins that work by interfering with nerve conduction in the same way that local anesthetics do. For example, the pufferfish produces something called tetrodotoxin, and that also blocks voltage-gated sodium channels, but it's not as nice of an outcome because that can actually cause death. Okay. And so the last little point here, summation. I know I've been talking about summation, but I wanted to take the time to actually discuss what it means and the two types, spatial summation and temporal summation. So spatial and temporal summation, with both excitatory postsynaptic potentials. Okay? So when we talk about postsynaptic, that means after the synapse. So I'm just going to draw this kind of like this, like a cell body. Okay. Postsynaptic means over here. If it's an excitatory postsynaptic potential, that means we probably opened up a sodium channel and we allowed a positive signal in. Okay, a depolarizing signal in. Okay. Inhibitory. So an inhibitory signal means that we probably let a negative charge in. Okay, so inhibitory postsynaptic because it's after the synapse, excitatory postsynaptic signal. That's all we're talking about with those things. They're kind of explained by the name. You just have to recognize the name. And then we're just trying to let that reach the axon hillock. Okay, so in order to do that, we have to have either a whole lot of signals come at the same time. Just one is probably not going to do it. So we might want to have a whole bunch of signals at once, causing depolarization. Okay, so if you can imagine, see these 3 neurons coming there. Of course, in real life, it's going to be, you know, hundreds or thousands. But if you have enough of them, you might get that signal to go all the way to the axon hillock. However, you can also have inhibition. When we have it in this kind of a situation where we've got 3 of them over space, kind of spread out, that's spatial. That's spatial summation. These guys, no matter where they come from, all decided, they could all come from different places. They've all decided that this neuron needs to be activated. So that's spatial summation. Now, let's say we're going to ignore the inhibitory right now. But this works with inhibitory too, the summation. But let's say that we only had 1 neuron, but it was very adamant, and it just kept repeating, saying don't go, don't, or no, it's saying activate, 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 activate. And it does it so much that you have a buildup of sodium in here that causes an action potential to make it all the way over. That's temporal summation. So spatial refers to how much space is being taken up. How much space is being taken up on the neuron, and then temporal is how many times, I guess, that it repeatedly activates to cause that to happen. Okay, so that's the difference. All right, and this is putting everything together. We've got that graded potential. So we have a signal that comes in that says depolarize. Okay, and then you have, it makes it all the way to the axon hillock. And then you have depolarization, sodium rushing out because a voltage-gated sodium channel detected it. So you have, it opens up to sodium, sodium rushes in, depolarizes, and then this repolarization phase is potassium rushing out. And then that causes your refractory period where it can't go again. Meanwhile, that depolarization signal has moved down to the next one. And it continues to do that. It can only move in that direction because of that refractory period. And then everything is just kind of pumped back into place, so you're ready for it to keep going in another round. Okay, so it moves, that action potential moves all the way down. It can split, bifurcate. It can split, and then you have neurotransmitter release at the axon terminal. Here you have a presynaptic inhibitory neuron that says, nope, not you, you're not going to do it. So there you've got inhibition and then neurotransmitter release. And that's the way it all works. That's the action potential. And then we'll talk about the synapses later. Thanks.